And as Jeff said, I oversee the clinical operations here at MAR-Bio. Really, my job is to make sure that you all and the patients can access our testing services and have a smooth process there. Just a little bit about my background. My background is in genetics as a genetic counselor by training and data science. And I previously worked at a genetic testing laboratory that was focused in postnatal neurodevelopment for over a decade. So I'm excited to talk about the MAR-Autism Test and share why I think it's an important piece to the multifactorial puzzle of autism. And I'd like to introduce Dr. Judy Van de Water, who is a professor of medicine and associate director at the Mind Institute at UC Davis, as well as our scientific founder. So we'll start our presentation first with Judy describing the science, and then I'll talk through the test itself and a little bit of information on how to access this test for your patients. So with that, Judy, I'll kick it off to you.
Thanks, Megan. Excuse me. So I think, we sort of began this journey thinking about what if we could predict which children would be affected by autism. And I think during the time when I started this around 2000 was the big Andy Wakefield stuff, everything that was going on with vaccines, which we've kind of come full circle back. But and I think that was the beginning of my journey. And I'm a classically trained immunologist in clinical immunology. And we thought about the immune system as a different way to look at autism. And the MAR autoantibodies are a potential pathogenic biomarker of autism risk. And these may actually define a targetable etiologic pathway in a subpopulation of women who have a child or are at risk for having a child with autism. Next. Just a little bit of background. It's, we are looking at MAR autoantibodies across the placenta, just like IgG does during a typical pregnancy. And these antibodies we have found are able to bind to their target proteins in the fetal compartment. This is anywhere for the research side of things. So in my lab at UC Davis, we've got a range from 12 to 26% of cases, depending on the population that we're looking at. We know that these antibodies change how neurons develop. In the pictures on the right there, I can't control my mouse to show you because I'm not controlling the slides, but the picture on the right, you can see both intracellular staining of these antibodies in a mouse model, and then how the neurons develop with a normal one on the left and a MAR exposed one on the right, with the lacking or immature dendritic spines. These are more severe kids. They have more severe behaviors, more pronounced stereotypic behavior, and we've noticed in both our clinical cases and all of our animal models, volumetric changes to brain. So these are the kids that are the largest brain kids that you may have heard about in autism. Next. So when we were first developing this, the Western blot on the left is what we started out with, and that's just fetal brain probed with samples from a mother of an autistic child, a typically developing child, or a DD child, and we have 3 in each group there. And we started to notice in the autistic sample population that we had antibodies to these particular bands that you see highlighted by the arrows. It would have been nice if it had been 1, but it actually turns out there are 8 different proteins that we now know are targeted by these antibodies. And it's combinations of 2 or more of these that give you that elevated risk. And these are highly specific, as you can see from the blot, and that has held up. They're very, very specific to autism. Next. So these, as I mentioned, the patterns, this is from research in a postnatal study. So this is associative data from a paper that we published in 2021. And we noticed that the patterns that you see, the combinations on the left, are the patterns of 2. We broke it down, or we winnowed it down to 2, that you need at least those 2. And you can see from the where it says TD total there, these are the patterns that are highly specific that we only saw in cases and not controls. And then over on the right, the odds ratios. So the odds ratio, they would have been very high because we're not seeing it in the controls, except that the number of cases that we had don't get us quite to that highest level of significance. But the odds ratio of 31 is very high. It's one of the highest that you can have in any of the autism testing paradigms for the biologics. Next. And so that was associative postnatal. And we did do it, while we don't test prenatally, but we've done several studies and have some ongoing now that where we're testing during pregnancy, because we really want to know if the antibodies are present during pregnancy, what is the outcome of the child? And so we took the patterns that we identified in the first study and we applied them to this sample set, which is the early markers of autism study at a Kaiser research study that I had with my colleague Lisa Crowan. And we tested for these same patterns if they held up. And so, and they did, fortunately. My 18-year-old diapered cat is trying to crawl on my computer. And we noticed that the frequency of a particular pattern was a little bit different in this population, but we still had a high degree of specificity. One note about the general population controls here, those are not validated as typically developing. They are simply controls that never sought services in the state of California. Next. So while we are convinced and feel very confident that the biomarker aspect of these antibodies is very strong, one of the important aspects to us was, do they have pathologic significance? And so we have done several animal model studies. I mean, with autoantibody, to determine autoantibody pathology, one of the first things we do is passively transfer the human antibodies in, and we did do that and got changes in the offspring. We've done it in mouse and in monkeys. We've done two different non-human primate studies in rhesus monkeys. But we felt it was really important to have these antibodies made by the animal that recognize the same parts of the protein that the clinical antibodies recognize. So we created a clinically relevant model where we create the autoantibodies in the dam long before we breed her, then we breed the dam, and all offspring and all births from all breedings will end up being impacted. We look at behavior, we've done imaging, we're looking at where these antibodies are in the brain, and then we're culturing organoids from these animal models as well. Next. And we, unlike genetic models, it does not matter what the genetics are of the animal that these are in, because it's all driven by the autoantibodies. And we saw that both mice and rats displayed altered social and self-grooming behaviors really similarly. And if we go to the next slide, that we saw a decrease in communication, definitely decrease in social behavior, which was especially apparent in the juvenile play, and increased repetitive grooming. We also, in our imaging studies in these animals, noticed we had some male and female differences in the way the brain responded to these antibodies in terms of volumetric size. However, that on the behavior side, there is no difference between male and female. So this, unlike we think of autism as a high male to female ratio, if the MAR antibodies are present, they are both impacted. Next. We also have done imaging in both the rhesus macaque passive transfer model and in our clinical population. And this slide just demonstrates that in the rhesus monkeys on the left, we saw again the same thing, enlarged brains, which on the right in our clinical population, the middle, the red graph is the MAR-ASD, where we have autism without MAR on the left and typically developing on the right. You can see these are the largest brain and a much tighter tighter volumetric size for the group. So that all of those data, from all of our animal models plus our clinical study, and we're about to launch another human study, tells us that there is a physiological impact of these antibodies on how the brain is developing, and that is leading to changes in total cerebral volume. Next. The social deficits in the monkey were interesting. I think monkeys have such higher order social behaviors that they're a great model. You can't do a lot of them because they're terribly expensive. But one of the things that we noticed that the mothers of the MAR-IgG treated the mothers of MAR-IgG treated offspring were very overprotective, which is definitely going to impact their social development. As they got older, the IgG treated offspring did show evidence of abnormal social behaviors with both familiar and unfamiliar peers. And the IgG treated MAR monkeys failed to engage in species typical reciprocal social interactions, meaning they would come up, they can be like, they would approach, so they would have social approach, but whatever behavior they were portraying was not seen as species typical by the non-impacted animal, and therefore was not reciprocated in a typical fashion. Next. So the main takeaways is, in the work that we're doing and have done that led up to this test, is gestational immune dysregulation may contribute to risk of altered neurodevelopment. We've got a lot of other studies going on looking at that. Maternal autoantibodies to these proteins in the developing brain definitely cross the placenta, because we've tracked that, and impair neural development. Similar to the processes we see in hemolytic disease of the fetus or newborn, or congenital heart block in SLE patients in their offspring. Large epidemiology studies support specific patterns of these antibodies, that they are predictive of autism risk and potentially the relative severity of affected offspring. And I can go into that more if you have questions. The antibodies localize to the brain, and we've shown that, and we can see them in specific brain regions. And the mouse and rat models capture all three domains of autism, in that they have social deficits, communication deficits, and repetitive behavior deficits. We also see the enlarged brains, as I noted, and then gestational exposure to these antibodies actually changes the brain cytokine and chemokine expression in the offspring, and not in the way that we thought it would, that it's elevated, but it's actually deficient. So all of those cytokine, chemokines, and growth factors that are necessary for healthy neurodevelopment are actually reduced in the MAR offspring brains. Next. So and this is my team. I have a phenomenal team that has done this work, and I'm happy to take any quick questions that we have on this part. And if not, Megan?
Okay, thank you, Judy. So I'll talk a little bit about how we translated that, more than 2 decades of research that Judy's been undertaking into a clinically available test, and then go from there. So I mentioned my background as a genetic counselor, and as Judy said, this is not a genetic test, so I want to clarify that. But there's a lot of overlap. And, as a genetic counselor working with families in the neurodevelopmental space, I experienced firsthand how often families talked or asked about autism likelihood. And really, they were just looking to best prepare for their family, whether that was family planning or access to services. And we actually surveyed over 300 providers who underscored this point, that they reported that women of children that they were providing care for often asked about autism risk. So what are the tools that are available to us to help inform parents around this risk? Well, again, come from a genetics background, and we know that multiple studies have shown that autism is highly heritable with strong familial clustering. And, of course, this naturally leads us to a high suspicion for a genetic etiology. But, I will say, even with the most advanced genetic tests available, we ran exomes, chromosomal microarray, Fragile X, genome. Uh, we did not find an underlying or identifiable genetic cause in the majority of the patients we tested. Yes, we have to still learn more, and these are rare conditions, but there seemed to be a missing piece here. This gap leads us to ask what other than genetics could be contributing to a recurrence in families? And what got me excited about the research that Judy was doing is, this shared gestational environment with these persisting autoantibodies in the mother could result in the increased recurrence risk that we see in these families. So the MAR-Autism Test, it was designed based off of that foundational academic research that Judy summarized. And it is a one single test that can be used in multiple different scenarios. So first, you can inform on recurrence risk in the preconception stage. You can help identify any younger siblings when they are either too young to be symptomatic or screened for autism. Or you can help to triage children showing signs of autism, and maybe they are on a lengthy diagnostic wait list, and families are looking for answers and potential guidance of what they can do to best care for their child at that time. So during our own independent clinical validity study at MAR-Bio, we took all the development work that was done in the academic environment and replicated and validated it actually in two CLIA lab environments. We identified the 5 combinations of these autoantibodies that, oh, sorry, my cat just stepped on Here we go. Okay. So we identified the 5 combinations of these autoantibodies that, when present in a mother, results in a very high risk of autism in her child. And these autoantibody combinations and test performance is remarkably similar to those that Judy just demonstrated, in the academic research. And this really highlights the robust underlying mechanism here for the MAR-Autism subtype. The test performance provides confidence in interpreting the results with a 97.5% positive predictive value and a high specificity. It was really important for us to minimize, the rate of false positives that would be received from this test, because important clinical guidance and decision making for families could result from it. And this is what's great is that the test only needs to be collected from the mother. We're looking for the autoantibodies in the mother that would then be passed on to the fetus during pregnancy. So there's no need to draw blood from the child, which I experienced in talking with many families in my past role, how a blood draw in a child could actually present a barrier for families pursuing testing. I do want to highlight that with this current version of the test, the mother must either have a child diagnosed or suspected with autism to be a candidate for the test. We are looking at expanding to more general populations in the future, but right now we are focused on that this first release of this version of the test in a higher prevalence population. And then finally, I do just want to highlight that while there may be some research that has been done in looking for these autoantibodies during pregnancy, the current test is not validated for use during pregnancy, although that is an area down the line that we will look towards. So, I want to talk about how a positive MAR-Autism test impacts the family, and really how do we frame risk for families. Well, as a genetic counselor, I would have conversation with families about risk. And a lot of times it was based off of observational rates in the population. So let's say a family presented to me, had no known family history of autism or etiology. They'd ask, what's the risk for autism in a child? And, the best we could really give them was some general population risk around 3%. Now, if a mother presented with already having one child with autism, well, then we would also quote similar kind of general risk of recurrence based off of observational studies. And that average was around 20%, plus or minus a couple percent, depending upon, the sex of the child first presenting, et cetera. Now, if that mother tests positive on the MAR-Autism test, that risk jumps to 97.5%. And that really provides a level of certainty that we haven't been able to provide before, apart from some rare syndromes that I've talked with families about in the past. Now, that's for the kind of preconception recurrence risk type of indication for use. Now, we do have the other use of the test, which is if a child is showing signs or symptoms suggestive of autism. And, a common screen for that would be an MCHAT to indicate that they're at, higher risk for autism to go on for a diagnostic. And that's around 58%, depending upon the study. Looking at the performance of that screening tool. So really, when you compare the information, the risk information that comes after receiving a positive from the MAR-Autism test, this is really impactful information. And it really can be a clinically meaningful number that a family can act on. And I'll say, in my background being in genetics, sometimes the best estimate we can give someone is a 50% risk. And that can seem, we don't want to minimize it and say flipping a coin, but, it could seem like that doesn't provide the certainty that families are looking for. And so this really is a meaningful risk estimate that comes from this testing. So how can a positive MAR-Autism test help families? Well, as I mentioned, it can help inform family planning decisions in the preconception period. And, I really like to think this could be considered alongside or, similar to carrier screening that's done in the preconception period. People are looking for their risks prior to pregnancy. And with this information, families can decide how that impacts their pregnancy planning. And, it may open up other pathways such as surrogacy, if that's something that would be available to the family. Again, understanding that it's the shared gestational environment that leads to the autism here. So surrogacy could be an alternate pathway here for them to pursue. Now, additionally, if a family is or a child is showing signs suggestive of autism, this test can help inform triaging and early intervention strategies. I've talked with plenty of families who expressed frustration at, having to wait a long time and feeling like they couldn't do anything. Having this information, while they would still be on a, potential wait list or go on to, obtain a formal behavioral evaluation for an autism diagnosis, they could use this information to maybe help start intervention strategies or educating themselves about autism that they could implement in their home environment. So finally, as I experienced as a genetic counselor, having an answer can be immensely impactful for parents. Again, reflecting in my past experience in genetics, there are a lot of genetic conditions that we couldn't provide direct medical intervention for. It was really anticipatory guidance and potential recurrence risk information. But I did find that having an answer, having a biological understanding could really help families move on from the why did this happen, particularly with some of the blame that could be felt by families, to moving to what can we do now? So identifying a reason can be really empowering and help families with their children, rising to their maximum potential. So, I really view this as a new tool in our toolbox. Again, back in my previous experience when families received an uninformative genetic test, there wasn't too much more that we could say to identify an underlying cause or etiology of the autism. But this is a new tool that can help us understand the mechanism behind autism in the child. And as Judy mentioned, Once we understand the mechanism, we can start understanding how there might be some effective treatments or mitigation of these autoantibodies and their effect on the developing fetus. So, I think this really opens up an avenue that's not only informative for families now, but helps us as clinicians and researchers identify potential treatment pathways down the road. So my next section is talking about how to access the test itself. But before I go into that, I just wanted to pause and see any questions, comments around the use of this test, the performance of the test. Yeah, Dr. Gabby. Oh, I can unmute you. There we go.
Turn on my mic. Hi. So I think in the beginning you said that you found the autoantibodies in 12 to 26% of cases. And how many mothers did you screen?
Oh, we have done, I think we're like up to close to 3,000 samples that we've tested in all our studies.
Great, good.
And
oh, go ahead. Yeah.
Well, I was just going to say, and Dr. Gabby, this is something that I do want to clarify. Our test, which again we went through our separate kind of clinical validity and development of the test, it has about a 6 to 8% detection rate. So it is a smaller and higher risk population that we're focusing on this iteration of the test initially. So we like to say we don't actually know the true prevalence of MAR-Autism, and it differs based off of the population that we're testing. But our initial results from the clinical validity indicates that we should expect a positive coming back in about 6 to 8% of cases initially.
Thank you. That's helpful.
Did you have another question, Dr. Gabby?
Are there other tests such as this autoantibody test for mothers with children with autism, or are they doing any genetic screening for autism in prenatally specifically?
I can answer for the other tests, not clinically, no.
Right. Okay.
Yeah. And I was going to say, the common screening tool in a preconception, we're going to say is carrier screening, which, is a battery of tests for specific autosomal recessive conditions that would have significant impact, of which some of those may have autism as a component of the syndrome. But not autism itself.
Right. Okay. Thank you.
I had a quick question.
Sure.
So you said that if they test positive for one of those combinations, it's a 97% likelihood that there will be autism. And if they test negative, what does that tell us? Like, can we assure them that they wouldn't have autism, or it doesn't mean that?
It does not mean that, because this is a subtype detection. What we're really looking for are these specific high-risk combinations that relate to MAR-Autism. And so we're not, it's not an all autism test. It really is a rule-in test for MAR-Autism. So we can't say, this rules out autism altogether. It does make MAR-Autism very unlikely in the children.
Okay, thank you.
Mm-hmm. And I see a raised hand. Is that?
Yeah. I have a question about, and I apologize if this was talked about when I stepped out, but is there any certain phenotype for kiddos with MAR-Autism that we should look for in terms of if there's younger siblings, or to know who should be referred for testing? Does that make sense? Does my question make sense?
Yes, it does. And I think there's two ways to answer that. And first, I'll answer from clinically what we know, but I think Judy has some insight from the academic side and research side of what we're seeing. Right now, I would say we don't fully understand the phenotype and how distinct it is from other types of autism. And so when you're looking at a family who may be a candidate for this test, certainly if they already have a child with autism, and that's where I kind of talk about this is another tool that can fit into your evaluation toolkit. So let's say, I would say in any family that you would do genetic testing for, trying to look at the etiology of autism, they would also potentially be a candidate for this type of testing as well, because it looks at a different cause of autism and can be, informative from that recurrence risk and just an answer standpoint. So, Judy, did you want to comment again on the potential phenotype?
Sure. Yeah, when we did the prospective study, the one that we tested during gestation, we noticed that there were some patterns that were very characteristic of kids with autism and intellectual disability, where other patterns were exactly the opposite, that they had a low incidence of the intellectual disability phenotype. So I think we also see in our animal models that there are differences in the behavior based on patterns. So it's more granular than just yes, autism risk, no. But at MAR-Bio, we're actually now, gathering more samples to test that in the clinical with the clinical test to verify all that. But there definitely is a phenotypic difference depending on which pattern they have.
And I will say that is an active area of research that we're interested in learning more about. Is there a particular presentation in that specific for MAR-Autism?
Yeah, I think also like if it tends to be related to co-occurring intellectual disability or the support level that kids typically need if it is the etiology.
Yes. And I think also in thinking about that for those kids, if you knew really early, how much could you mitigate some of that, the impact of those, deficits?
Yeah. It's exciting.
Okay. And Dr. Botcher, I thought you had your hand up.
Yes, part of my question was that actually, thank you for this presentation, and it's exciting to have a potential biologic marker. I think that's what we're all looking for. I guess my my big question and concern is really, so what are we expecting from this in terms of phenotype, and because autism is such a spectrum, and there's mild to severe, and then you wrap in the intellectual disability. Which is a whole different sort of profile, and so that I guess that's what's going through my mind right now is okay. So if this test is positive, then what do we tell mom that yes, the child has a higher risk of having this type of autism, but then what to expect from a just practical, phenotypic, and what do we do about it? Because again. I have loads of patients in my practice that have that they have diagnoses of autism, and they're so varied in their ability, and autism is so diagnosed, behaviorally that again, in my 30,000 years of doing this, there's many different autisms, and I do believe there is one that you've described, but again, the phenotype would be a certain profile, right? And so anyway, that's what I was really asking about is how do these children present, and do you have pretty consistent presentations? Are they all very severely intellectually disabled, or are they fairly higher functioning that you can
I can
address
honestly
it in a certain way?
Yeah, I can honestly say that these kids are more impacted. These are not the Asperger's kids. These are which you wouldn't diagnose this young anyway. The kids in all these studies were like 2 to 5. So, but I think that is definitely in my research and as a company, one of our biggest sort of interest right now is identifying, because if I tell you this information, I would love to also be able to tell you, and this might be what works for these kids, right? Or this is what the trajectory of this child might look like. And with intervention, could we change that? Because if you can test really early, you have, you know, as I'm sure you know from your clinical practice, you have a better chance of, while the brain is still plastic, at least having some potential impact. We are also, as a company and in my lab, working on some, you know, therapeutic in terms of blocking these antibodies from even having access, right, to the fetal brain, so that we can mitigate, you know, just the chance of this this particular autism. So I think, you know, it's a long road to get to do all these things, but we've got the animal models, which are helping us with that part of it, because the models are really good in terms of replicating. And we've learned a lot from looking at the brains of these, the exposed rats in just the downstream. So we've done some RNA sequencing of the brains. There are a whole host of things that are going on, just that we're changing in the brain just because these antibodies are present, that I didn't anticipate. It reduces the cytokine chemokine levels below normal. So one might think you'd have inflammation, but it's actually the opposite of that. We've got a deficit, which those factors are super important in healthy brain development. So I think I'm thinking just immunologically of the things that we might be able to support that might help, not just blocking, but all the things that you might be able to do postnatally to support healthier brain development. Sorry, that was a long research answer.
No. I mean, I didn't expect, to be honest, I didn't expect, you know, a clear-cut answer, because I don't think there is one.
I don't think we have one yet. No.
No, I don't think we do, and I think that's what I'm really struggling with is if you have this test, what then? So it's positive, and then what in terms of the next question parents ask is what should I be expecting from my child?
Or
and
what can I do? Right?
Yeah. What can I do, and what should I expect? And you know, that's the question I get very often. If my child has autism, now what? And parents are always looking ahead. They want to know, well, will my child have what kind of life will my child have? And that's so that I'm struggling with this. You know,
yeah, no,
what do you tell a parent that may have a positive test and really they're at a crossroads in terms of deciding what to do. And again, I don't know at what stage in the pregnancy prenatally can be done.
During pregnancy, actually.
It's preconception, you said.
Preconception or any time during the postnatal.
Got it. Yeah.
And there's a couple of reasons. I mean, the assay is not validated for, pregnancy physiology and changes, but also to that point, because we still are learning about this, we want to be, very cognizant of how that information can be used for pregnancy decisions. But, I kind of go back to some of my experience in talking with really hundreds of families navigating the kind of diagnostic odyssey. And I've talked with families who had a child who's, 5 years old, diagnosed, they're in the, intervention pathway, and, the family just had a brand new baby and they're worried at 3 months. What should I be looking out for? What can I do to help recognize this earlier? Is there anything that I can do if, I do start to see concerning signs? And, I think this could also be a tool that could be empowering in that, and maybe help reduce some of that watching and waiting anxiety that families can experience in that scenario. But, each family is different. And, I think this is going to be a different tool and different potential value depending upon where they are and their different experience. So.
And one other thing, Dr. Butcher, I wanted to add, I think since we aren't general population testing yet, the families that would have this test would be those that have been already exposed to autism in their family. A lot of the people that are interested right now in talking with us are those that have an autistic child diagnosed and are curious about the risk of having another child. And then also, because a lot of times by the time their child's diagnosed, they already have another child who's maybe 12 months or 18 months old, and they're curious as to, the risk in that child as well. So, they may not know how severe their older child is going to be at that point. But now they'll know that this other child's following that same trajectory if it's positive. For those children that are not yet diagnosed and symptomatic, I think that's kind of where you're probably referring to is like, okay, they're positive now, but how impactful is it going to be? And I think that's I'm not sure in your case if that's your use case or not. I think one of the interesting things we've run into is typically, when you have a test like this, you're communicating with specialists where the child itself is the one that's getting tested. In this case, it's the mom. And I believe in your case that the mom's not your patient. And so, but what's happening is the providers are telling us, well, the mom's asking me because she's in there with the child. And so what do I do? And I think Megan will get into this later, but we have a telemedicine option available because there's some providers who are maybe not able to have the mom be their patient or enrolled as their patient and so on and so forth. So there's a lot to it. We recognize that. And as Judy alluded to, there's a lot more that this company is going to hopefully do in terms of preventing it from happening in the first place. This is just kind of the first step. So your questions are consistent with others, I will say.
And how do we know that if the first child has autism, that they have the same subtype of autism?
I mean, that's a great question. I think I can tell you if the mother is positive, subsequent pregnancies will also be impacted. But if you get her tested between the two kids, you don't, right? Because we don't know when they arose. All that we can say at that point is that they're associative.
It's
Good
tricky.
questions.
It's
What's that?
tricky. Said
It
it's
is.
tricky.
Autism
I'm
is tricky
really
enough,
struggling
but
with
yeah.
this.
Yeah.
Yeah. Yeah, I think it brings up, questions again that we're actively interested in gathering evidence for is why are these autoantibodies present to begin with in the mothers? What is that potential trigger for these to be present? We have evidence that they are durable, persistent for potentially decades in some patients. But yeah, I mean, that is a good point, Dr. Batra, that, yeah, I mean, there is a point of when we don't know when these autoantibodies arise, but we can look at the statistics and the correlation from the various studies to look at, how this lines up with the occurrence of autism in the families. We have about 10 minutes remaining. These are all great questions. And like Jeff said, all questions that we've, really good questions that we've had from a number of physicians. And it just reflects that this is although there's multiple decades of research, it is a newly recognized, subtype. And there's a lot of areas that we would like to investigate to better understand and help better inform both providers and families about this condition. Just briefly, I do want to talk a little bit about process to accessing the test. So we have set up a portal. It's an online portal where a short order form is completed. You can choose to either have a blood draw conducted in your office, or if that's not something that you routinely do, we're happy to coordinate it with the patients directly at a lab location in their area. And on the patient side, we contact them to complete their authorization for the testing and payment online. And then once the specimen is received at the lab, the test results are delivered to the portal electronically about 14 days after the receipt of the specimen at the lab. The test results, we've tried to design it in a way that is straightforward and clear regarding what the results mean. So I know the left view is small. We can send a sample report out for you. But what I wanted to highlight is the language, the interpretation language around a positive or a negative. And this kind of goes back to the question of what do you say in the case of these different type of results. In the positive result, we say this indicates a 97.5 risk that current or future children have or will develop autism due to these maternal autoantibodies. And then in the negative, we say there's an absence of these, significant combination of these autoantibodies, and that this indicates a low risk that current or future children would be affected by autism due to MARA. But a negative result does not exclude all causes of autism. And we actually go in and explain that further in the clinical consideration section of the report. We want to make sure that both clinicians and families are very aware of the rule-in nature of this test. So finally, this is a brand new test on the market. It is currently offered as a self-pay test. It is $640 if the test is paid in full. There's a prompt pay discount for the patient, or if the patient chooses to split the standard cost of the test into 4 payments over the span of 4 months, the total cost would be $985 if they choose that path for themselves. So that was the information about the test and how to access it. I know we do have a few more minutes for questions, and I know, Jeff, you wanted to get the poll out there. But I did just want to say, we're a small team, but a dedicated team. We've all come to this company because we're really excited about the research that went into the development of this test, and really the opportunity that this new biological mechanism for autism has to help with the therapeutic approach that really doesn't exist in a lot of autism cases today. So myself and our clinical science liaison is available to answer any questions that you have. We are also available to speak with families, although it is more on an education standpoint, and we would direct any clinical questions or clinical guidance to the providers for those type of conversations.
Are you marketing this with other physicians besides pediatricians, like genetics clinics and OBs and things like that?
So interestingly, Dr. Gabby, since we've just initially launched, we've been talking to specialty types of all kinds. Anybody who sees patients who have autism or mothers who have children with autism. So we've been talking to OBs, MFMs, dev peds, ped neuros, pediatricians, functional medicine, integrative medicine, IVF, endo. It's quite a wide range of folks, to be honest. In many cases, there's folks who may have, very few patients this will be applicable for. And others where it's extremely applicable. There's a lot of developmental pediatricians and ped neuros. They feel like they're diagnosing the child, and maybe this test isn't quite ready for them yet. But there's like pediatricians, they have referrals to some providers where there's long wait lists, and they're interested in trying to triage for them. So we're still narrowing down what makes the most sense, but realistically, we're trying to educate people on its availability. There's patients that have reached out to us directly who have children with autism and are curious about proceeding with other pregnancies and want to get tested before they do so. So it's quite a wide ranging. And if you guys have any questions, obviously you have our information or Mariah's information. She can contact us to answer any further questions you guys have. I hope everybody has a good evening.
Thank you.
Thank you, guys.
Thank you very much. This is great.
Thank you, everyone.
Thank you. Very interesting.
Bye.